Hello everyone, I am Feroz Ahmed, a graduate student from Bol Lab of Department of Biology, and welcome to my poster session. I work with Acinetobacter baumannii, responsible for hospital-based infection and can rapidly develop resistance against multiple antibiotics. And this bug can infect different locations of human body, can cause ventilator-associated pneumonia, bloodstream infection, urinary tract infection, septicemia, and many more. Carbapenems are typically used to treat multi-drug resistance pathogens. However, more than 60% of hospital-acquired E. baumannii infections are resistant to carbapenem treatment. And CDC says these carbapenem-resistant E. baumannii are most urgent threat to public health. And World Health Organization prioritized E. baumannii as a critical for new antibiotic development. Since E. baumannii infections are increasingly failing to respond to carbapenem treatment, physician has to go back to the last resort therapeutics, colistin, to treat carbapenem-resistant infections. And interestingly, these bacteria possess a unique capability to turn on biosynthesis of LPS, which is considered as a universally essential component of all gram-negative outer membrane and used as a target site for colistin action. In figure 1a, we can see that absence of LPS allows the bacterium to survive colistin-mediated killing. As a graduate student, I'm curious to find out how E. baumannii maintain the stability of the outer membrane in absence of LPS. Here in figure 1b, I'm showing the RNA sequence analysis which determine the altered expression profile of RNA transcript of LOS deficient cell compared to their parent strain. And we noticed that increased expression of putative lipoprotein in LOS deficient E. baumannii. And therefore, my hypothesis is that lipoprotein synthesized in the cytoplasm transported all the way to the outer membrane and remodeled the outer membrane to provide integrity of the LOS deficient gram-negative cell envelope. In figure 2, I'm showing the proteomic analysis of peptidoglycan attached proteome. And we found out one putative lipoproteins LPP1 were attached to peptidoglycan in wild type but not in delta LDTK, which is a putative LD transpeptidase responsible for cross-linking glycan chains of peptidoglycan. Additionally, two lipoproteins LPP1 and LPP2 were present during this outer membrane stress but not in wild type. And interestingly, both of these lipoproteins LPP1 and LPP2 has characteristic C-terminal lysine residues, a motif essential for LD transpeptidase dependent covalent attachment of the lipoproteins to the peptidoglycan stem peptides. And lipoprotein mutant E. baumannii failed to recover any LOS deficient colony like wild type, which suggests the importance of these lipoproteins in respect to the integrity of the LOS deficient E. baumannii. In neuropeptide profiling, using HPLC further validate the role of LDTKs in lipoprotein attachment, is showing in figure 3. Neuropeptides are digested with trypsin that leaves the trilysine residues, and we have seen the presence of trilysine peak in wild type but not in LDTK mutant and delta, and also lipoprotein mutants. And in figure 4, we evaluate the integrity of the membrane in absence of lipoproteins. Excessive shedding of outer membrane vesicles means an unstable outer membrane, and I saw an increased outer membrane vesicle production in lipoprotein mutant relative to their wild type, which supports a model that lipoprotein attachment stabilize the outer membrane. Additionally, in figure 4b, we evaluate the permeability of the lipoprotein mutants using ethidium bromides and noticed increased permeability of outer membrane lipoprotein knockout strains compared to the wild type strain. So, altogether, we concluded that this physical tethering of this outer membrane lipoprotein to peptidoglycan increased the cell envelope stability in response to outer membrane defects. With that, I would like to convey gratitude to my advisor, Dr. Joseph Vole, my lab mates, collaborators, and fundings, and a special thanks to Discover Committee for the opportunity to showcase my research. Thank you very much for listening to my talk.